Okay, let's go. The review, chapter 2. 외국 학생들 밥 사준다 그랬어? 그래. Chapter 2 and chapter 3 review. Any problem or questions? You can look up the review session. Any question or the multiple choice question that you don't know the answer or confusing? Some plants live in bogs in which the pH is about 2. Thus, these plants are able to survive blank external environment. Basic, buffered, acidic, neutral, alkaline. Which one? pH 2 is what? Acidic, right? Okay. Chapter 3, we go through carbohydrate, lipids, proteins, and nucleic acid. Any questions? 읽다가 이거 잘 모르겠는데 설명 다시 우리말로 설명 필요한 거? 응? 뭐? Any? No? You know everything there? Okay. So number 2, chapter 3, question number 2. Functional groups often confer the general property of blank on blank molecules. A. Hydrophilia, water-based. Hydrophobia, water-based. Stability, carbon-based. Stability, unstable. Polarity, carbon-based. 지금 아무도 없어? Anybody print out the summary? 그래서 다른 사람들은 다 모르는구나. Okay, what is it? Do you remember we went through functional groups, 작용기, functional groups. Okay, that gives what? That gives polarity, right? Charged group. Polarity to what? Carbon-based molecule. 이거 챕터 끝날 때마다 풀어야 되는데 안 그러면 나중에 중간고사 볼 때 너무 많은데 공부해야 될 부분들이 챕터 끝날 때마다. So you should read, every chapter after the class or before the class would be ideal, but it's not easy, right? So after class, during the 추석, you have to read. Okay? 그럴 거야? Okay. So let's go chapter 4. Okay, chapter 4. So life's home, the cell. We saw the movie, right? So we are composed of cells. Okay, how many cells? Trillions of cells. Okay. So life's fundamental unit is the cell. Okay, so this chapter 4. 3단원 안 끝났었던가? Where were we? Sorry. I have memory lapse. Nucleic acid. All right. Do I have the slide? Yes. All right, nucleic acid. Okay. So it says acid. So there must be something can donate H+. Okay. So again, nucleic acid is also composed of from small building blocks to long chain. Okay. What do we call that small building block of nucleic acid? We call it nucleotide. More words. Okay. Nucleic acid in Korean, hexan. Okay. Nucleic acid has two kinds, DNA or RNA. Okay. Both cases, the building block is called nucleotide. Okay. We'll see how they look like. Okay. So the nucleic acid, DNA and RNA. What is the difference? D and R. Yes, good. DNA stands for deoxyribonucleic acid. So DNA and RNA both are nucleic acid, right? The only difference is they use different sugar. They have different name, okay? The name of sugar that they used in DNA is called deoxyribose, okay? The sugar that used in RNA, it's called ribose. That's why it's called DNA and RNA, okay? We'll see the difference, okay? So what is the component of, what are three components of nucleic acid, DNA or RNA? Okay, three components of nucleotide is phosphate and sugar and base. Okay, one more, phosphate, sugar, and base. In this case, this is DNA structure. Okay, they call it DNA double helix. 이중 나선 구조. Okay, it's called DNA double helix. Okay, 1953, Watson and Crick they come up with this model of double helix, and they got the Nobel Prize. So Watson was, when he was discovered, he was 25 years old. He just got his PhD in US and went to United Kingdom, England, and he was doing his postdoc. And his partner, Crick, was a graduate student in physics department, was a little bit old graduate student, about 30-something. And they discovered this structure, okay? The structure is double helix. You can imagine their staircase goes like this. Have you been to a lighthouse? 등대 가본 적 있어요? 등대? Or if you on board battleship, 전투함 같은 데 올라가 봤어? 거기 계단들이 so the stairs in those narrow places goes like this, right? If you can imagine those staircase goes around like this. So the handrail of that staircase is this one. Phosphate and sugar is the backbone of this double helix, okay? And then steps, okay, steps, 발판, okay, is composed of this base. We call it base, okay? What are three components of nucleotides? Sugar, phosphate, base. Now, one more step. Base has 4 different kinds, okay? We can stand for G, A, T, C. Okay, those are 4 different base found in DNA. Okay, they are found in DNA. Okay, so together, going back, DNA, you start with these 3 components. Okay, sugar, phosphate, base is one nucleotide. Okay, these nucleotides strung together, you will have DNA strand, one strand. The other strand is the same. Okay, the only thing is in the middle, these 2 bases faces each other. Okay, we call it they are pairing each other. Okay, by what? These little dots. What is this? These little dots. What is this chemical bond? Do you remember? There were 3 chemical bonds, right? Covalent bond, ionic bond, hydrogen bond. That's the little dots is hydrogen bond. So base G and C are connected by hydrogen bond. A and T is also connected by hydrogen bond. Do you see the difference? No? Yeah. G and C has 3 hydrogen bond. A and T can have 2 hydrogen bond. Okay, next question. So if you, let's say, you started to boil this DNA, okay, then what will happen is this double-strand DNA will go like this. Okay, then the question is, if one strand is all consists of AT, 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 the other strand is consists of GC, 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 GC. Okay, which one will have higher melting point? GC, because they have more hydrogen bond. You have to add more energy to break it, right? So good. All right. So we understand this. DNA is this structure. Okay. This structure is very, very important. Okay. As soon as they published this double-strand helix, 8 years later, the Nobel Prize was given. Okay, because before the structure, they was wondering how this genetic information is exactly copied to next generation. They couldn't understand. But once they saw this structure, it is intuitively clear, because if you open this strand, okay, and each strand can be used as a template, and they can copy exact opposite strand. We'll come back to that point, okay? So the structure is very important. DNA usually form this double helix structure, DNA. RNA has more flexible structure, okay? DNA is more stable. So most of living organisms store their DNA into more stable structure, which is DNA, okay? But the advantage of RNA is they're flexible, is changing fast. That can be also an advantage or disadvantage, okay? A good example is viruses. Virus can store their genetic information in RNA. That's why they are changing very, very fast, okay? That's advantage and disadvantage. But, all right. So DNA, we understand DNA consists of nucleotide. Nucleotide consists of phosphate, sugar, base. The name of sugar in this case is called deoxyribose. That's why we call it DNA. Deoxyribose, nucleic acid. Why acid? This phosphate group, do you remember functional group of phosphate group? There's 1 P and there's 4 oxygen, right? And many of them can be negatively charged. Okay, so question? All right, DNA is a repository of genetic information. The sequence of its base encodes the information for the production of huge array of protein produced by a living thing. What do you mean? What is the information stored in this DNA? Is the sequence, G, T, G, A, C, those sequence, what comes after? So they have 4-letter language, okay? DNA has 4-letter language, G, A, T, C, or, okay? RNA has a little bit different language. They use G, A, U, C. Instead of T, they use U. That's the only difference, okay? So the one base is a little bit different, okay? But they're structurally similar to T. So just substitute T with U, then it's become language of RNA, okay? All right, second nucleic acid is RNA, okay? The only difference is they use different sugar called ribose. Okay, that's why it's called ribonucleic acid. That's why we call it RNA. Okay, which transport the information encoded in DNA in its site to the site of protein synthesis structure called ribosome. That's getting difficult. We'll cover this chapter at chapter 4. Okay, just come with me. Okay, so we went through 4 molecules that are important for life, okay? The first one was carbohydrate, 탄수화물, right? Carbohydrate. We talked about 4 big carbohydrates, okay, that consist of small unit, which is monomer, which is in this case glucose, okay? 2 of them were storage of energy. The other 2 was structural function, okay? lipid, they don't have a structural, they don't have common structure, okay? So we talked about triglyceride, steroid, and phospholipid, and wax, okay? And protein consists of amino acid. How many kinds of amino acid there are? There are 20 different amino acids, okay? In protein, the structure is important, right? There's primary structure, secondary structure, tertiary structure, and quaternary structure. The last one is nucleic acid, okay? There are two kinds, DNA and RNA. The difference is they use different sugar, okay? The building block of nucleic acid, we call it nucleotide, which consists of sugar, phosphate, and base, okay? Okay. Questions about nucleic acid? All right. All right, so with possible exceptions of viruses, all the cells, all the life forms on Earth is either a cell or composed of cells. In other words, it's called cell theory. In other words, every living creature consists of cells. The first video we saw, our body consists of trillions of cells. But it wasn't clear until someone looked at it, okay? We cannot look at with naked eyes, so someone invented microscope, okay, around 17th century. At the end of chapter, you can read about Leeuwenhoek, started. So if you look at closely inside, they start to see a unit in our body. That's we call it cell. Okay? So what about viruses? They don't form a cell, so we don't define them as a life form, but they are alive in a way, right? They can infect, but we don't count as a cell, and we don't count as a living thing. Okay? So cells come into existence only through activity of the other cell. What do you mean? Cells are coming from cells, same thing. The life form comes from life. What about the first cell? We don't know how it come up with it. Okay? And all the cells can be divided into two types, prokaryote and eukaryote. Okay, it's like it's like living place. We don't have that kind of apartment in Korea, but apartment called studio. Okay, if you rent this place, studio, it's a just open space. They don't have any room. Okay, it's like prokaryote. Prokaryote, inside the cell, there's no separate spaces. Okay, in a way, it's easy. You can eat, you can sleep here, you can watch TV here. But if life gets a little bit more complex, you want to have your own space. Okay, you want to have your bathroom, you want to have your room, you want to have your kitchen, the compartments. You want to divide the spaces. Okay, now that's the eukaryote. Okay, so the first thing eukaryote decided to is they want to keep their genetic information into safe place, which is nucleus. Okay, we call it HEC. Okay, so the difference between prokaryote and eukaryote is whether they have nucleus or not. Okay, prokaryote is called 원핵세포 또는 원핵생물. Before nucleus. Pro means before, karyot is nucleus. Okay. Eukaryote means real. Eu means real. Okay. They have nucleus. That's why we call it 진핵생물 in Korean, in Chinese character. Okay. So the division between prokaryote and eukaryote, whether they have nucleus or not. Not only nucleus, but also eukaryote start to have their own small compartments, spaces. Why? It's more efficient in a way. Okay, you can all the cooking in kitchen. You can go to the bathroom, take a shower. So the space can be specialized into certain purpose. Okay, so that's the advantage of eukaryotes. Okay, so all cells can be classified as prokaryote and eukaryote. Okay, so what are prokaryotes? Okay, there are bacteria, and another group is called archaea. It's type of bacteria, but its meaning is old bacteria, archaea. Okay, so this is division of all the living things. Okay, so these two, bacteria and archaea, both of them are, we call it prokaryote. They don't have nucleus. So the cell is kind of open space inside. Okay, the rest of it we call it eukarya. Okay, which means they are eukaryotes, 진핵생물. Okay, so there are 4 groups in eukarya. We know animal, plant. Okay, but there are 2 other groups. This one called fungi. In Korean, it's called 균류. Okay, what are they? Mushrooms and what is it? What other? Fungus. Yes. Memory lapse. Yeast and fungus. Okay, if you look at mushrooms, they look like plant, don't they? Right, but don't say mushrooms are plant. Okay, that's no. Okay, why? They don't do any photosynthesis. They don't make their food. They're sipping their nutrients from the dead animals or plants. Okay, and some of the proteins the mushroom make is more closely related to animals than plant. Okay, so the fungi is more close to animal than the plant. Okay, they are not plant. The 4th group in this case is called protist. In Korean, it's 원생생물. Okay, so this is every single. It's not a good grouping, but the living thing, they don't belong to plant, they don't belong to animal, they don't belong to fungi, they belong to protist. That's the definition of protist. Okay? All right, so now we're moving into from prokaryote into eukaryotes. Okay? Eukaryote cells have most of their DNA contained in membrane-lined compartments called the cell nucleus, cell nucleus. Okay? Whereas prokaryote cells do not have a nucleus. That's the defining definition difference between prokaryotes and eukaryotes. All right? All right, so summarize the whole thing. Okay, you start with prokaryotes. There are 2 groups, bacteria and archaea. Okay, the rest of it we call it eukaryotes, 진핵생물. Okay, there are 4 groups: animal, plant, fungi, protist. Okay, so this is another picture. So in other words, why they put it protist here? Okay, they don't have better classification. Just put everything here. Protist can be as different as from each other from animal to plants. So this is the protist. Okay. So more practical difference between prokaryotes and eukaryotes. Prokaryotes, they don't have separate space inside the cell, but eukaryotes, they have nucleus. They have also other compartments too. Usually, prokaryotes are smaller than eukaryotic cell. Okay, there's good reason for it. Okay, and usually prokaryotes are single cell. Bacteria is a good example. They can stick together, but it's just a single cell. Okay, but eukaryotes, it's multicellular, means our body, now they start to differentiate. In other words, heart is concentrated in beating function, and the other tissues, our skins, is protecting from outside. They have different roles in the body. Okay, so they have multicellular differentiated cell. Okay, only inside the cell, what do they have? They have only one type of organelle. Organelle means small organ. In this case, it's called ribosome. We'll come back to that point. Okay, in eukaryotic cell, there are many small compartments and small units inside. Okay, that is different. Question? Okay, we'll move on. Okay. All right, so we'll go through eukaryotic cell. Okay. 생각보다. So if you look at page 67, 5th edition, 4th edition, 65, there are, in the bottom of the page, 65, there is 5 components of eukaryotic cell. We'll go through one by one, okay? So the nucleus, okay? So you can compare the whole cell as a city of soul, okay? It's a big, okay? But, you know, if you go inside the cell, it's big, okay? So the nucleus is the control center. In other words, city hall maybe. All the official business is done in city hall. Okay, so that's the nucleus. Second one is plasma membrane, is the boundary of the city. Okay, so usually when you're coming in Seoul, you don't see checkpoints, no guards, right? But if it is a country or, you know, you're going in and going out, there's check place. Okay, if you enter some other country, they will guard. They will ask you to show passport. Okay, so that's the boundary, the plasma membrane. Okay, and then third one is called cytoskeleton. Skeleton means bones. Okay, cytoskeleton. Cyto means cell. In other words, the structure of the cell. Okay, in other words, constructions. You can build something, you can, you know, destruct something. That's the cytoskeleton. Okay. Then the number 4 is a cytosol or cytoplasm. Same thing. Okay. You know, it is the space between boundary and outside of nucleus. Okay. And then last one is organelle. Organelle means small organs inside the cell. There are several of them. Okay, so now we'll go into one by one. Okay, so this figure, where are we? Yeah, page 68 is a little bit kind of intimidating, right? But we'll go through one by one. So again, this is the nucleus. Okay, inside we keep DNA. Okay. Sometimes we see DNA as a form of chromosome, but not always. Okay, we'll come back to it. Inside the nucleus, there's another round structure called nucleolus. Nucleolus. In Korean, that's a in. Okay, so don't worry about it. Yeah, don't worry. Just remember the nucleus, the bigger one. Okay, that's the control center. All the information is stored in there. There is a membrane, okay, structure we called Where are we? Endoplasmic reticulum. Difficult name. Endoplasmic reticulum. In short, we call it ER, emergency room. No, not. Okay. But just for you to remember. Endoplasmic reticulum says ER. ER has two types. One is smooth, the other one is rough. Why is it rough? There's small little dots you see here. Okay. Those are the ribosomes. That's where the workbench for the protein synthesis will come back to that point. Okay. Now the outside is plasma membrane, the boundary. Okay. Then there is a few organelles, small organs inside the cell. Okay. This one look like, what does it look like? 번데기 같이 생겼는데. It look like mitochondria, right? Look like cocoon, whatever. Yeah, mitochondria, and there is the stacked membrane. Looks like McDonald's pancake. Okay, it's called Golgi complex. Okay, and then there's a little round shape called lysosomes. We'll come back to each of what their function is. Okay. Okay, so the cytosol. Okay, cytosol. Okay, so a little bit. It's just. It can be as a space between nucleus and inside the membrane. Okay, cytosol or cytoplasm, same thing. Okay, space or the fluid, gel-like fluid that fills this space. Okay, that's what we call cytosol. Okay, then cytoskeleton. Okay, is a construction material. Okay, there are three types of ropes. Okay, we'll come back to that point. Okay, it's a little bit hectic, right? Okay. Okay. One more. Okay, so 5 components of cell. Okay, the nucleus, the controlling center. Okay. Other organelles will go one by one. Mitochondria, Golgi complex, lysosome, in plant cells, chloroplast. Okay. We'll just focus on animal cell at this point. Okay. Cytosol is a space or fluid filled between this nucleus and inside plasma membrane. Okay. Cytoskeleton. Okay. It's a skeleton means bones. Okay. So they are the constructions inside the cell. Okay. The last one is plasma membrane, his outside boundary. Okay. Boundary of the cell. Okay, now we go into look at the cell, but in the base of the question is how you look like your parents. Okay, the question goes. Okay, then you have your parents' DNA. Okay, what information is in DNA? Okay, we'll see, and that information somehow has to be come out and show. Okay. So that information is stored in DNA, and that is being copied into RNA. And based on that information, you will make protein, okay, that will express yourself. All the protein is expressing yourself, okay. All these things happen inside the cell, okay. We'll follow each step where inside the cell this thing happening, okay. So we'll go. This figure. So page 69, so this is how the protein is being made inside the cell. Okay. So we store DNA in where? Inside the cell. Where do we store DNA? In nucleus, yes. So DNA, we saw double helix, 2 strands intertwined together, right? They located in nucleus. Okay? But in order to make protein, okay, you cannot take out DNA from the nucleus because it's important. You want to keep it in nucleus. Instead, you want to make little copy of it, and then you bring the copy out. Okay? That's the function of RNA molecule. Okay? In this case, small m, messenger RNA. Okay? The messenger has the message about the information. Okay? So what do you call it? You make RNA from DNA. Well, okay, so you copy one side of DNA into RNA, the yellow strand. This RNA coming out of nucleus. Okay, this is nucleus. Now you're coming out of to where? You come out of nucleus. Where is it? Inside the cell, outside the plasma membrane. What do we call it? Cytosol or cytoplasm. That's the space or the liquid that fills in, right? So the RNA came out as cytosol. Okay, you can have two places to make protein. Okay, one of them is just cytosol. Okay, so oops. So the RNA comes out, stay here. That's okay. That's in cytosol. Okay, but they have to have this little thing called ribosome. Okay, it looks like little, but it's a big protein and lots of protein and lots of RNA inside. We call it ribosome. Okay, this book says it's one of organelle, small organ. Okay, so RNA has to meet this ribosome to make protein. Okay, so one place it will meet is cytosol. Okay, the other place in this case showing here is membrane compartment. Okay, membrane. There is a narrow passage by membrane. We call it endoplasmic reticulum, ER. Okay, there are two types of ER: smooth ER, rough ER. Smooth ER, they don't have this ribosome, so they don't make protein. But rough ER, why is it rough? All this ribosome sticks to this endoplasmic reticulum. Okay, that's why it's called rough endoplasmic reticulum. Okay, so RNA comes out, they sits on top of this ribosome. Okay, and they start to make protein. Okay, in this case, these proteins are supposed to pack and send out of the cell. Okay, so in other words, they come to this place, post office, to send this protein out. Okay, that's where this protein goes. So the Golgi complex, this is post office, 택배 place. Okay, you can ship these proteins out of cell. Okay, how do they pack it? They pack it in the vesicles. They wrapped around with membrane. You saw the movie, right? Something comes in, the membrane wraps around it. It's going out is exactly the same. You have to wrap it with the membrane, and it will go out of cell. Okay. So in other words, this Golgi complex is a sorting place. Okay, sorting place for protein. Okay, and then finally, it will go to the membrane and goes out of cell. Okay? 너무 어려운가? Is it okay? You follow it? Some, not all of them. I know it's a little bit difficult to follow. Okay. A few more organelles and then we'll have break, okay? Just bear with me, okay? Okay, again, this pancake, stacked pancake structure, we call it Golgi complex. What a name, okay? Why do we put capital here, Golgi? As someone's name, someone's name who first look at the microscope. This looks strange structure. I'll put my name on it, so it's become Golgi complex. Okay. Again, it's a sorting place. In other words, 택배 place. Okay, the protein comes, they put label. This goes to somewhere. This goes to somewhere, and they wrap it around. Okay, that's the function of Golgi complex. The second organelle explained in textbook is called lysosome. Don't get confused with ribosome. Okay, L and R. Lysosome is recycling center. Okay, it's if old thing comes, they'll break it down. Okay, and then make a new, or whatever. They break down old things. Lysosome. Why? They have very good enzymes inside. They can cut everything. Okay, so it's recycling center. The only thing wrong about this figure is you cannot make mitochondria out of scratch, which means you cannot start mitochondria from small pieces. Okay, just like the cell, mitochondria comes from mitochondria. Okay, so what will happen is if you comes little things, there should be one mitochondria. It will uptake, and it gets bigger and split into two. Okay, to see the difference, you cannot make from scratch. It's called de novo synthesis. It's a very difficult word. De novo synthesis means there's nothing before, and then you make one. Okay? We know that's not possible for cell and living things. Same thing with organelles. They don't make mitochondria out of scratch. Mitochondria always split from mitochondria. Okay? That's the wrong thing about this figure. Okay. So why mitochondria is important? We'll come back whole chapter of it. Okay? Mitochondria is the power plant of the cell. Power plant, it's like cash machine. Okay? So they will generate ATP. Okay? They'll generate ATP, which is cash. Okay? Inside the cell. Okay? So that will give you lots of energy. So if you look at the cells, if it is muscle cells, you see lots of mitochondria, because when you move, when you run, when you do the exercise, the muscle cells need a lot of ATP. Okay. Now the next one is called cytoskeleton. What is a cyto? Cyto means it's about cell. Okay. Skeleton means bones. Okay. So this is a construction inside the cell. Okay. There is three types of ropes inside the cell. Okay. 3 types of ropes. Okay. Based on thickness, we classify it as first one, microfilaments, intermediate filaments, fine, right? Small one, middle one, the big one also called micro, but it's a tubal. It's a hollow big rope. Okay, again, microfilaments, intermediate filaments, microtubule. Okay, the thinnest rope, middle one, thickest rope. Okay, what is the function of it? First one, intermediate, is most stable one. Okay, they don't change very much. Okay, this microfilaments is very actin. It consists of actin protein. You don't have to know about it, but it's microfilaments. Where do they use? Like this. This is big macrophage, your defense system. Okay, if bacteria invade from outside, they can eat them. Okay, they can eat. They try to eat bacteria here. How do they eat? They can protrude a little bit of hands, not hands, but foot. It's called pseudopod. Okay, and that's the trajectory goes. That's little ropes is inside. Okay, they can go out, or cells can go move like this. Okay, those are by these microfilaments. Okay, so next example is microtubules, the thickest one. Okay, this one. Tubules, microtubules. Okay, it's a hollow in the inside. It's empty inside, but it's the thickest rope you can have inside the cell. What is the function? It has three functions. Okay, we saw in the movie they can be subway rails. Okay, inside the cell, it can transport little things. Okay, so this is microtubule, and there is a train can go and come. Okay, so microtubule is transport. Okay, there are two other types. They can form tiny hair called cilia. Little hair. If you cut in the middle, you will see this one. Okay, so microtubule can form cilia. The third one is a long whip. Okay, whip. Okay, you can see in the tail of sperm, it can swim with this flagellum. Flagellum is singular. Flagella is the plural. Okay, so cilia, flagellum, and this transport is made up what? Microtubule, the thickest rope inside the cell. Okay, all together, what do we call it? Cytoskeleton. Okay, cytoskeleton. Okay, almost there. Right. The last part of chapter is comparing plant cell versus animal cell. So far, we looked at the inside of animal cell. So we'll see just the difference between plant cell. Okay. Plant cells have something that animal cells do not have. Okay, what are they? Cell wall. There's another boundary. Okay, and then there's a large central vacuole. If you think about fruits, all the juice is stored inside the cell. Okay, the central vacuole stores it. The third one, organelle called chloroplast. Because of this chloroplast, plant cell can do photosynthesis. In other words, with the sunlight, they can make sugar. Okay, how nice. What if we have chloroplast in our cell? We just go out and in the sun, you don't have to eat lunch. It would be very nice, but you can. Okay, so this is the plant cell. So the cell wall is outside support, outside of cell membrane. Okay, then there is a central vacuole inside, storage, and also they function as a recycling center. Do you remember in animal cell, there's a small organelle called lysosome is doing the same function. Okay, that's why plant cells don't have lysosome. Okay, because central vacuole can have that function. Okay, now the plant cell has this organelle called chloroplast. We'll look at the chloroplast. Okay, there's some structures. There's outer membrane, inner membrane. We'll come back. This is chloroplast is site of photosynthesis. What is photosynthesis? 광합성. Okay, photo means light, and synthesis, you're making something. Okay, you make sugar using sunlight.
The scientists sometimes make a convenient division of eukaryotic cells into two types, animal cells and plant cells. These cell types have more similarities than they do differences, but they are different enough that it will be helpful to look at them separately. These are the main structures in an animal cell and their primary functions. Endoplasmic reticulum, assembly line. ribosomes, workbenches, mitochondria, powerhouse, lysosomes, cleaning crew, cytoskeleton, structure, Golgi complex, distribution center, nucleus, control center. As noted earlier, plant and animal cells have more similarities than they do differences. Among these similarities is that plant cells do most things that animal cells do, that is, produce proteins, transform energy, and so on. There is only one structure present in animal cells that plant cells don't have, the lysosome. Plant cells have some features that animal cells do not have. A thick cell wall, a large structure called a central vacuole, structures called plastids, one important variety of which is the chloroplast.
3 more minutes, okay? So we looked at one cell inside, right? But we're, as a human, we are multicellular organism. In other words, we have lots of cells, okay? Cells is the fundamental unit of it, but it's not separate from neighbors, okay? They have to somehow communicate, okay? So how do they communicate? Okay, there's 2 structures you have to remember. In plant cells, it's more difficult because, to remember, they have very thick cell wall, okay? So you have to have some space between 2 cells. We call it plasmodesmata. Names are killing you, right? But you have to bear with it. Okay, animal cells, a little bit, easier. So it's called gap junction. It's not gap t-shirts, but it's gap junction, okay? Gap junction. Did you see the movie at the beginning? And all the muscle cells start to twitch, right? If you grow cells together, they start in one bit. They twitch in one bit. Why? They are connected, okay? So the ions go through it, and then they have this pacemaker. They got the rhythm, okay? So cell can communicate each other through this type of structure called, in plant cells, it's called plasmodesmata. In animal cells, same function is done by gap junction. Okay, I think that's about it. Right, so the whole summary of cell, and we'll have like 10-minute break. And if you're sleepy, you can have some chocolate. And one chapter is called Matthew effect. Okay, Matthew. This is from the Bible. Okay, so to everyone who has, more will be given, and then he will grow rich. But from the one who has not, even what he has will be taken away. 있는 사람은 더 받을 것이고 없는 사람은 있는 것마저 빼앗길 것이다. Sounds unfair, right? Yes, it's unfair. Okay. So he started the stories in Canadian Ice Hockey League. In Canada, the ice hockey is their national sport. Okay, everybody's crazy for ice hockey. Why? During most of the year, they're extremely cold. Usually, they don't go out. They indoor ice rink they start to. So kids like 4 and 5 start play. Okay, so what happened? So the big game in Canada, the ice hockey team plays. Okay, so someone looked at their birthday, the date of their birth. Okay, it turned out 70 or 80% of players are born in January, February, March. The question is why? Okay, so the kids league for 5 or 6, 7 years old league, they start gathering these kids from January 1st to December. Okay, if you're born in January, at the age of 4, you are a little bit bigger than the other kid who was born in November or December. A little bit bigger, a little bit more developed. Okay? And then we can consider him as a better player, and we'll select them. And they give them better coaching and better camp. They start to play better, and they make the national team. Okay? So same thing true for soccer team, national team. So he goes on like this. So the success seems to be, people think if you work hard and if you're determined, you do it and you will make it. But that may not be true. Okay? The society and your environment and everything around you is important. Okay. Also, there is an interesting case. Okay, this one is called Langdon versus Oppenheimer. Okay, so we think if you do success, you must be very smart, right? So this is an example. There's two people, Langdon and Oppenheimer. Langdon is very, very smart person. Okay, his IQ was immeasurable. In other words, IQ test 190 or almost 200. Okay, and what happened to him? He dropped out of college. Okay, the other extreme case is Oppenheimer. He was also genius as a kid. Okay, so he goes to his PhD in physics. He goes to England. Okay, and he didn't like his professor, advisor. Okay, there was a trouble between them. And he tried to poison him. And he got caught. And what happened? He wasn't expelled from the graduate school. He received his training and then came back to the United States. And he was one of the key members in Manhattan Project. Manhattan Project is the project that developed atomic bomb. Okay, so what is the difference between two geniuses? One genius dropped out of the college. The other one caught in criminal act, but he got away from it. Right? You have to read the book. Okay, so this is a recommendation. And also there is an interesting story about Korean Air crash story. Okay? All right, chapter 5, it's about plasma membrane. It's a little bit short chapter, so we can finish a little early. Good. Okay? So the nature of the plasma membrane. What is the plasma membrane? It's outside boundary of the cell. Okay? It's important because it can control what's coming in and what's going out. Okay? So plasma membrane is a thin, fluid entity managed to be very flexible, okay, but yet it's stable enough to stay together. All right, so do you remember one of the type of lipid called phospholipid, right? Phospholipid is the major component of membrane. So we call it membrane as phospholipid bilayer. There's 2 layers of phospholipid. Okay, we'll see that. Okay. Second molecule in membrane is called cholesterol. Do you remember? This is type of steroid. Yes, steroid lipid. Okay. We see cholesterol. If you have high cholesterol, it's bad for you, but you need to have cholesterol. Okay, because they are membrane components, plasma membrane components. Okay, and also there are some proteins sitting in the membrane. Okay, and the last part is glycolysis, which is, do you remember glycoprotein? What is glycoprotein? Protein has a little bit of sugar in it, right? Sugar attached. This is glycolysis, is the sugar part of this glycoprotein. Okay? It looks like this. Okay? There are 4 components. This is phospholipid. Okay? They draw as a red head and 2 white tails. Okay? This is one molecule of phospholipid. They form 2 layers of it. Okay? This is membrane. This is the boundary. Okay? And there's some cholesterol is in the middle, and then some proteins attached to it. Okay? And glycocalyx is sugar part of this protein. Okay? So those are the 4 components of plasma membrane. So the first thing is how this phospholipid form this structure. Okay? Do you remember? The phospholipids start with what molecule? Start with glycerol, right? Glycerol has 3 carbon, has 3 OH group, and 2 of them are attached to fatty acids, right? 2 of them attached to fatty acid, and the third one attached to phosphate group. Okay, that give you charge and also polarity. So it become polar means they like water, hydrophilic. Okay, these 2 tails, okay, because they don't have anything else but carbon and hydrogen. Okay, so they are nonpolar, they are hydrophobic. So if you mix this phospholipid with the water, what happened? Okay, this side is water. Inside the cell, outside cells, everywhere is water. Okay, in our body. So this head part is facing outside and facing inward because they are hydrophilic. Okay, this part is hydrophobic, so they are away from the water. Okay, that's how the membrane forms. Okay, so this is outline of cell. Okay, think about some polar molecule, charged molecule. They cannot go through it. Okay, because they cannot pass through this hydrophobic part. Okay, but some molecule can do it, such as steroid hormones. Okay, they can go pass right through it. Okay, some of the hormone works that way. They don't have any membrane receptor. They just can go through it. Okay. All right. Second part is cholesterol. So what is the function of this little thing, cholesterol? Okay. So cholesterol, you have to remember one thing. Cholesterol molecule, okay, keeps optimal fluidity. In other words, cholesterol level determines how fluid is your membrane. Okay, if you don't have cholesterol, maybe it gets rigid. Okay, but if you put some cholesterol inside, it can become fluid. Okay, so now third component was protein. Okay, so it can be said everywhere in our cell, all the function is carried out by protein. Okay, so protein can have many different functions. Okay, there are 2 types of protein on membrane. Okay, one of them is like this one, which is embedded in the membrane very deep. We call it integral membrane. Okay, where do we have it? Here. It's better. Okay, it's the protein that goes all the way through the membrane. It's called integral. Okay, another type is just touching the membrane, just barely touching membrane. We call it peripheral. Peripheral. It's just like English class, right? Peripheral and integral protein. Okay, there are 2 types of protein. But this protein can have many different functions. They can have 4 different functions. Okay? Number 1 is structural function. Something you have to have anchor. You have to have anchor into the membrane. Okay? Do you remember the macrophage is trying to eat the bacteria to use cytoskeleton? You have to put something, right? But you have to have some place to hold onto it. Okay? That's the support. Okay? That means anchoring. Okay? Those are membrane, those are protein that anchors into the membrane. Okay? And then these little things, it's cytoskeleton from there. Okay? So in other words, what is the function? It's structural support. Okay? Second one is recognition. Okay, recognition. Okay? Which means in our body there is trillions of cells. Okay? And also the defense system of our body is always surveillance. In other words, they patrol. Okay? They keep whether the virus is coming or bacteria is coming. So one way to do that is cells, normal cells, okay, put some piece of protein and then they make a flag out of it, okay, and then put in the membrane saying, I'm your side, okay. Then this army goes through it, and these are our side. They don't attack, okay. But if virus is infected or some bacteria is infected, okay, those cells chop up some of the viruses and then present to the outside of cell, telling, I'm infected, so kill me, okay? Then army go through it and kill it, okay? That part is called recognition, in other words, immune response, okay? Some of our disease, atopic skin, or things are kind of allergy, those are too sensitive immune disease. In other words, even if you're on the same side, your defense system is attacking yourself. Okay? So its balance is very, very important. Okay? So recognition. Okay. Third one is communication. In other words, let's say some of you still are growing in size, right? So growing means cell is not going bigger. Cell has certain size, so what do you have to do? Cell will divide into 2, 4, 8. Okay, that's growth. Okay? So how do you know you have to grow or you have to stop growing? What is the message? Some of hormone tell you, right? And hormone from your brain maybe and send a signal. And those signal goes to the cell and then come to this receptor. Okay, they can fit into this receptor. Those signals saying you have to divide, you have to grow, or you have to stop growing. Okay, and that gives signal, and that can be recognized and communicate with the cell. And inside the cell, this signal goes and then say, we have to make something, we have to make more protein, we have to divide soon, those kind of communication. Okay? And the fourth example is transport. Okay? Because this membrane is a barrier, okay, you cannot easily go out and come in. Okay? So what do you have to do? You make special passage for those molecules. Okay? Transporters. Okay? So molecules can go through. Okay? So there are 4 membrane protein functions. Okay? Support, recognition, communication, and transport. Okay? Not difficult, right? All right, we'll go. So next one, we have to understand diffusion, gradients, The last one we want to know is osmosis. Okay, osmosis. Diffusion is an easy one. If you drop a drop of ink into the water, what happens? At the end, it will like this. Okay, so it can be understood as diffusion. The little ink molecules concentrate here and then start to disperse. Right? That's a diffusion. Okay, diffusion can be defined as from higher concentration to lower concentration, molecules go. Okay, or thermodynamically, they want to have higher entropy or something like that. Never mind. Okay, so diffusion from higher concentration to lower concentration. Okay, higher concentration to lower concentration. At the end, it will go like this. So the question is, can you go back from here to here? You can go back, but you have to put energy into it. Okay, how can you do it? You maybe filter it or somehow dry it and then come up with again. Okay, but diffusion is from higher concentration to lower concentration. Okay, so the difference between concentration, we call it gradient. Okay, gradient. Okay. So now, what is osmosis? Osmosis, 삼투 현상. Okay. Osmosis is the net movement of water across a semi-permeable membrane. So in other words, osmosis happens in certain condition where the membrane is semi-permeable. What it means, semi-permeable? Some can pass, some cannot pass. That's our cell membrane. That's why osmosis is important for life phenomenon. Okay? So it's things like this. If you put semi-permeable membrane, which is the water molecule can go through because it's small, sodium ion or chloride ion cannot go through because they're bigger. Okay? Then what happens? If you put the same level of water, but then you put salt in one side. Okay? What happens is now you have higher concentration of sodium ion and chloride ion, right? So if it is normal case, what happens? Sodium ion and chloride ion go this way, right? From higher concentration to lower concentration. But because of this membrane, they cannot go. Then what happens? The water molecule from opposite side comes this way. Okay? That's the osmosis. Right? Okay? So what happened? It goes like this. Water goes like this. Okay, that's the osmosis. So that happens in actual cell. Okay? There are three solutions. You remember solution? Solution is solute plus solvent, right? So solution, there are three types of solution. Hypertonic, isotonic, hypertonic. Sounds all the same, right? Hyper means hyper is higher, okay? Hypo means it's lower, okay? Iso, we talked about isotopes, right? It's the same, okay, same element with different numbers of neutron, okay? So isotonic means the concentration is the same, okay? So in other words, if you put animal cells or plant cells into isotonic surround, isotonic means they have same salt concentration, okay? And nothing happened because going out, going in, water molecules are the same, okay? They're in equilibrium, okay? But if you put in hypertonic solution, think about making kimchi. Okay, you put cabbage into what? Salt solution, right? What happened? All the water comes out of the cell and the vegetable will shrink, right? because the water is coming out of the cell similar to, similar to this one, okay? But in hypertonic solution, what happens? Water molecule moves toward it, okay? So in this case, animal cell shrink, and also plant cell shrink. What happened? Plants wilted. They need water, okay? But in the opposite case, hypotonic solution, in other words, there's just water outside, okay? Our cells have about, what, saline, a little bit of salt in it, right? So what happened is water is moving in from outside, okay? So what happened? Animal cell cannot hold anymore because water is coming in, okay? At the end, they'll burst it, okay? But in plant cells, they don't because they have cell wall, okay? The water comes in, they can push it back, okay? So they can be swollen, but they don't go and burst, okay? So, okay? So that's the osmosis.
The plasma membrane, which is an animal cell's outer boundary, has 4 main functions. The first 2 are to keep what is inside the membrane concentrated and to keep harmful materials out. The third function of the membrane is to control the movement of many molecules into and out of the cell. The fourth function is the critical activity of cell signaling or communication. A double layer or bilayer of phospholipid molecules. Each hydrophilic head faces outward toward the watery environment that lies both inside and outside the cell. The hydrophobic tails of each molecule point inward toward each other. Cholesterol molecules act as a patching substance on the bilayer, keeping some small molecules from getting through and helping the cell maintain the optimal level of membrane fluidity. Proteins are either integral, meaning bound to the hydrophobic interior of the membrane, or peripheral, meaning not bound in this way. Membrane proteins serve 4 main functions. Structure support. Proteins may be attached to parts of the cell's scaffolding or cytoskeleton. Recognition. Binding sites on some proteins help to identify this cell to other cells, such as those of the immune system. Communication. Receptor proteins protruding out from the plasma membrane can be the point of contact for signals sent to the cell via traveling molecules, such as hormones. Transport. Proteins can serve as channels through which materials can pass into and out of the cell. Glycocalyx. These chains attach to communication or recognition proteins and serve as their binding sites. The glycocalyx can also lubricate cells and act as an adhesion layer for them. Diffusion is the movement of substances from regions of its high concentration to regions of its low concentration. The difference between the high and low concentrations of a solute in a medium is called the concentration gradient. Just as the natural tendency of an object is to move from a higher elevation to a lower elevation, a molecule or ion has a natural tendency to move from a higher concentration to a lower concentration. Solutes can move up a concentration gradient, but only with the expenditure of energy. Consider 2 containers of an aqueous solution that are connected by a tube, as illustrated at left. In the tube is a semi-permeable membrane. Water can move freely through the membrane, but solutes cannot. If more solute is put on the right side of the membrane than the left, more water flows into the right chamber, which has a greater concentration of solutes in it. Water molecules that are bonded to the sodium and chloride ions that make up salt are not free to pass through the membrane to the left chamber of the container. The net motion of water across a semi-permeable membrane from a region of lower solute concentration to one of higher solute concentration is known as osmosis.
Okay. All right. Are we there? No? Oh my God. We have a lot more. Okay. I guess we cannot finish this one. We only halfway through. Wow. But chapter 4 and chapter 5, homework. Okay, I only printed 30 of them because you can download print. So here and here. Okay. So next week we'll start from 5.3, moving smaller substances in and out. Wow. It's not easy. And anybody having problem going into online class to print out homework or print out your summary? Nope. So from next class, I will not bring any prints of homework. Okay? So you go to the online, download, print, solve the question, puzzle, and hand in next week. Okay? Any questions about today's class? I can answer, when we met next week, Tuesday, 11. If the weather is nice, probably we'll meet outside. I'll give you cell phone message. 네? 번개 야외 수업, maybe. Okay, and also foreign students, you are invited for lunch, so you can stay behind the class. I will see you next week. Okay. Happy 추석.